Hello, my name is Isabel Hubert, and today I will be presenting an overview of 3 fascinating research posters I attended that explore the ecological impacts of human infrastructure, particularly on the U.S.-Mexico border barrier, and on local wildlife populations in the lower Rio Grande Valley of Texas. These studies investigate how the installation of the border barrier disrupts habitat connectivity and impacts species such as the Texas indigo snake and collared peccaries. I'll walk you through the main questions, methods, and results, and conclusions from each presentation, as well as my reactions and connections to the material. The first poster I reviewed was titled "Don't Fence Us In: Evaluating Ecological Impacts of the U.S. Border Barrier System on Texas Indigo Snakes" by Samantha M. Dillion and colleagues, all graduate students from the Texas A&M University systems. The study addressed how the border barrier impacts Texas indigo snakes, a species that relies heavily on riparian corridors for movement. The main question explored was, how does the installation of the border barrier affect habitat sustainability and connectivity for the Texas indigo snake? To answer this, the researchers used species distribution modeling techniques, the SDM techniques. They complied occurrence data, connected field surveys, and used environmental variables like soil type, vegetation cover, and elevation to predict areas of suitable habitat. Then, they analyzed the effects of the border barrier on habitat connectivity. The results showed that most suitable habitats were located near the barrier, with forested lowlands being the prime areas. This led to fragmented habitats, especially in riparian zones crucial for snake movement. The ongoing research aims to assess how barrier installation disrupts dispersal routes and to identify the potential mitigation strategies. I found that this study especially was relevant to my interests in how infrastructure affects wildlife. The use of SDM was an innovative approach to quantify habitat loss. It made me think more critically about large-scale human projects that can disrupt species' natural movement patterns. This research connects to my studies in wildlife ecology, where we often discuss the importance of habitat connectivity. It was surprising how much the snake's habitat is directly adjacent to the border barrier, highlighting the urgent need for more research in these areas. The second poster was titled "Habitat Use and Movement of Collared Peccaries Associated with the U.S.-Mexico Border Barrier System" and was presented by a team of researchers including Way A. Rydberg and others from the Texas Park and Wildlife Department. The study focused on collared peccaries, a medium-sized ungulate, crucial for seed dispersal and as prey in the ecosystem. The main question was, how does the border barrier impact the movement and habitat use of collared peccaries? To investigate this, the researchers used GPS collars to track the movement patterns of peccaries across the areas with and without the barrier. The data collected revealed significant changes in their movement corridors, with barriers caused the reduced habitat connectivity. Preliminary results showed that wildlife crossings integrated into the barrier were being used by the peccaries, but they were also observed using agricultural lands near their natural habitats, which suggested a level of adaptability. I was intrigued by the use of GPS technology to track the peccaries, as it provided detailed, real-time data on their movement. The findings about wildlife crossings were encouraging, and it was interesting to learn that peccaries are using agricultural lands in addition to their natural habitats. This raises questions about the long-term sustainability of such behaviors, especially considering the challenges of maintaining these fragmented habitats. This research ties into border conservation strategies we've discussed in my wildlife management classes, particularly regarding the balance between preserving natural habitats and managing human impacts on wildlife. The third poster I attended was titled "Population and Spatial Ecology of Collared Peccaries in the South Texas Plains" by Abigail N. Dwelly and colleagues from the Texas A&M University systems. This study aimed to understand collared peccary population dynamics in southern Texas, an area that is critical to the species' survival. The primary question here was, how do factors like habitat sustainability, environmental conditions, and human activities influence collared peccary populations? The study employed multiple methods to gather data, including camera traps, roadside surveys, and helicopter surveys. GPS collars were placed on 64 collared peccaries, and researchers also collected the data on nutritional and health conditions. Over the course of the study, they gathered valuable data on the species' movement patterns, population density, and habitat across the 450,000 acres of land. The results showed that collared peccaries prefer dense vegetation for cover and food, but they also adapt to agricultural landscapes, raising concerns about the long-term viability of populations in heavily modified areas. The research is ongoing, with the goal of developing predictive models for population growth and habitat sustainability, which could inform future conservation efforts. I was particularly impressed by the comprehensive approach used in this study. The combination of camera traps, GPS collars, and helicopter surveys gave a holistic review of the peccary population in Texas. It also reinforced the idea of collared peccaries, while adaptable, might struggle in areas with extensive human modifications. This connects to what we've studied in population ecology, where environmental factors like habitat quality and fragmentation directly impact species survival. The potential for applying this research to other wildlife species in similar habitats is exciting, and I look forward to seeing how these findings could go guide future wildlife management strategies. In conclusion, these studies provide valuable insights to the ecological impacts of the U.S.-Mexico border barrier on local wildlife productions. They underscore the importance of considering habitat connectivity and fragmentation in conservation efforts, especially for species like Texas indigo snake and collared peccaries. The use of innovative methods for such GPS tracking, camera traps, and species distribution modeling offers exciting possibilities for wildlife research and management. These studies highlight the complex relationships between human development and wildlife conservation, and the need for thoughtful approaches to mitigate the effects of infrastructure on wildlife populations. Thank you for watching. I hope this report provided you with a deeper understanding of the ecological challenges facing the wildlife in the lower Rio Grande Valley and the innovative research being done to address them.